Hey guys, this is Yu Yang Ye, or you can call me Wilson. Today I'm going to talk about the last part, which is the discussion and conclusion of our article, A Novel Bacterial Protein Family That Catalyzes Nitrous Oxide Reduction. So for the conclusion summary, actually the paper makes a simple but important claim. There's a third nitrous oxide reducing enzyme family, L-NOZ, which is clay 3. It's a new finding. We've mostly been missing when we count how nitrous oxide gets removed from the environment. The authors show that L-NOZ genes are more broadly distributed across the taxa and habitats than older surveys suggested. So which means our picture of the global nitrous oxide sink may be incomplete before. The strike genomic pattern is that many L-NOZ clusters lack the unusual NOS accessory genes and instead sit next to the MACB ABC transporters and cytochromes C or B. It's hinting at the different maturation or electron delivery pathway. If that is true, there are two things follow. Many field assays that target only canonical NOZ likely under-detected this clade, and the climate model that rely on those data may undercount biological nitrous oxide removal. Here's a core message, a bigger but more diverse sink than we thought before, with clear implications for methods and modeling. So basically, I just introduced the discussion and conclusion of our article. Now, I really want to talk about something really beyond the discussion and conclusion. So first one is, did this paper follow modern article architecture? I'm going to put out the intro, which is the flashback of the article. It starts explaining why nitrous oxide is a big climate deal and highlighting the mismatch between what's emitted and what's being removed from the nature. Pretty quick, the main finding. The authors claim out the unrecognized nitrous oxide reductase family, which is a clay 3, aka L-NOZ, is definitely distinctive from the canonical clay 1 and 2. Here are core evidences. In acidic microcosm, the enrichment of SEP5, the powerful tool for nitrous oxide reduction despite few canonical NOZ reads, hey, this is a new type. Also, the key methods being used in this article, which is metagenomics, proteomics, and qPCR, they both link to the L-NOZ abundance to the nitrous oxide consumption. Last is the discussion impact. By redefining where global nitrous oxide gets removed, it's being way more broader than initial thoughts by the authors. Next identifies other players, microbes. They also play crucial roles to eliminate nitrous oxide. Last is a key finding, which has been combined and contributed to biotech and models to change the climate. So now I'm going to talk about the useful information to the field. Despite there are 4 key points across this slide, I kind of shortened them into 3 key items that are being immediately actionable. The first one is function. The L-NOZ as a third enzymatic solution for nitrous oxide reduction by increasing the functional diversity and likely resilience of the sink under changing conditions. The second one is distribution. L-NOZ appears widely across phyla and environment, indeed, which means surveillance tool must be updated. Canonical NOZ primers will miss it. However, the Bayesian abundance maps and model inputs also. A third one is mechanism leads. The recurrent MACB ABC transporter and also cytochrome B and C neighborhood, they are all near around L-NOZ suggest that it's a non-canonical maturation or electron transfer route. That's a strong evidence and testable hypothesis for genetics of biochem. When taken together, these points actually gave the field a concrete the to-do list, right? Redesign primers or the panels to see clay 3, kind of run head-to-head clay assays under realistic chemistry and probe the flanking genes to confirm the proposed pathway. Next is, what would I do differently? If I'm the author, what am I going to do in this case? If I were leading the next phase, I would push on the four fronts. The first one is field trials. Basically, by applying this crucial clay 3 L0Z to wastewater treatment plants or manual lagoons and rice paddies. By replicating and randomly of applying it, trying to find out and observe which mode is favored by L0Z, which can make the most efficiency of eliminating the nitrous oxide, turning into the harmless gas nitrogen. The second one is determine key traits. By set up the experiment, knocking out MACB, ABC transporter, and knock out cytochrome B and C respectively one by one. If L0Z stops working only when these are missing, they are actually part of the pathway, which means they're actually playing the crucial rules when regulating of the pathway of the L0Z. The third one is health impact modeling. By combining the nitrous oxide reduction and population health metrics together via climate changing, for example, crop yield impacts affect nutrition globally, and then make the model to calculate whether health co-benefits of nitrous oxide controls alongside of the cost, which means put the cost aside, ignore it first, just talk about the efficiency and the influence made by the nitrous oxide reduction. Lastly is the side-to-side comparison. I think basically test all three clays under different conditions. The first one is copper level, oxygen level, and also the measure how fast they turn the nitrous oxide into the nitrogen gas. And then knock out and restore strains, which is confirming which clade is doing the specific work, and use qPCR kind of to track them respectively and identify how clay 3 beats clay 1 and clay 2. Basically kind of take a look of the back to the phylogenetic tree, especially the clay 3 share with bunch of the ABC and MACB transporter, and also tons of the cytochrome B and C genes. These unique traits might be the crucial traits could beat clay 1 and clay 2. So now I'm going to talk about the future direction and the limitation. What are we going to do next? Actually, our short-term goal is turn this enzyme finding into real public health action. First one is apply it. Run a small pilots in farms and wastewater sites to boost L0Z microbes and measures nitrous oxide emission along with the soil chemistry and the community DNA, so we can show that clay 3 actually cuts emission in the real world. Second one is enzyme engineering. Make L0Z stable and efficient for use in bioreactors or let's say biofilters as the wastewater plants and agricultural sources. So in this case, we can see its real efficiency and the real function. The third one is monitor and model it. Use clay-specific primers, for instance, 1, 2, and L0Z clay 3 in monitoring networks, and feed those data into models. try to predict where we can reduce nitrous oxide the most. Now I'm done talking about what do we do next. I think it's the time to talk about the limitation. Actually, we're missing the few things that matter for public health. We don't yet have the field level proof that L-NOx-Z is actually causing the drop in nitrous oxide um, in the real world. The proposed MAC-B ABC transporter plus cytochrome gene pathway is promising, but not proven. And actually, many surveys still missing CLE3, which is schools, maps, and policies. So by fixing these gaps, we'll let cities and health agencies estimate the co-benefits, which is fewer ozone, related breathing problems, less heat stress, and try to yield more crops, and compare those benefits to the cost. That's how the science moves from the lab to the real world application to actually gaining the health, measurable health. Thanks for watching. That's all. See you next time.